Okay, thank you. Just I try to provide you briefly overview about this new drug because UV5008 is a new drug. It's a first-in-class reversible non-covalent epi-inhibitor developed for B-cell malignancies. I would like just to explain in which sense I call it this drug as epi-inhibitor, because when we start our study, first of all, we recognize one of the activity of this drug. This drug born as an epi drug, the epi inhibitor, the HDAC and the NMT inhibitors, but we see that in particular cancer system in which there are kinase aberration, we see a strong antitumoral effect. So we realized that maybe there are other properties that we don't know about this drug, and we see that. And we discovered that our drug is a double mechanism of action as anti-tumorigenic drug. One mechanism is the ability of this drug to act as a BTK inhibitor. The enzymatic BTK inhibitor. The second activity is the ability to work as an epidrug. In which sense? Is able to, in the first case, undergo inside the cells. This drug is metabolized, is a prodrug, is divided in two different active molecules by a cleavage at the disulfide bridge. This active molecule enter in the kinase pocket, block the BTK, and block BTK in a particular way because it's not involved in this inhibition, the cysteine 481. And we will know that these specific residues is an important residue for the development of resistance to the covalent BTK therapy. This is the first point. So this drug is able to bind the BTK and inhibit BTK kinase activity in a very strong way, non-covalent way. It is another important point. So we see that we're able to block the protein activity, but we're able to reduce in a very strong way. We in some way we completely abolish the expression of BTK, the gene expression. So also obviously the protein is dramatically reduced together the phosphorylated form of BTK, but also the total amount of BTK is completely reduced. So this is the important and the very very nice activity of this drug. We call it epi-inhibitor just to underline the double mechanism of activity of this drug. And obviously we compare during our study our drug with the conventional therapy that now it's present for the patient, the covalent inhibitors such as ibrutinib and so on, but also with the pirtobrutinib because it is our reference for non-covalent BTK inhibitor. Okay, and in the comparison, we verify that we have an additional activity respect to the pirtobrutinib because we have an additional ability to dramatically affect also the amount, the expression of BTK. Just this. And obviously, I would like just to add that the importance of our drug is the possibility to use this drug also when the patient develop the mutation in cysteine 481. This is another important point. Our reference pirtobrutinib have the same activity, and we well known as a non-covalent BTK inhibitor is able also to use and FDA approved just in 2022 for the relapsed and refractory CLL patients. And we hope that also our drug are approved and after the different phase of clinical trial arrived for this type of a patient. Obviously,